0764. So today in this video, I will show you the demonstrations and also explain to you a little bit about the principle of streak plate method and gram staining experiment. So, streak plate method were used by scientists to dilute or reduce the number of microorganisms so that they can obtain a single colony of the bacteria. And now I'm going to show you how streak plate method are conducted.
First of all, you are going to need a marker pen to draw two lines across the petri dish to create a four section. So before we start our streak plate method, we have to take the sample from the test tube, and the first step you need to do is to flame your inoculating loop, and then you have to flame the neck of the test tube before you dip your inoculating loop into the sample. And as a precaution, you need to flame the neck of the test tube again before you close the lid. So, after the sample has been taken, gently poke the surface of the agar and drag your inoculating loop across the surface to create the first streak. Before you go for another streak, make sure to close the lid of the petri dish to prevent any contamination and reflame your inoculating loop. Drag one corner of the first streak and then drag it several times across the second area to create the second streak. Make sure to not let the inoculating loop touch any of the previous streaked area. For the third streak, do the same procedure as the second streak. But for the last or the fourth streak, there is no need to reflame the inoculating loop.
So next, I'm going to brief about gram staining experiment, and I will show you the demonstration later. So for gram staining experiment, scientists use this experiment to differentiate between two types of bacteria, which is gram positive and gram negative. So in the supposed result, it will show you two colors of two stain of bacteria, which is purple violet and red or pinkish stain. So the purple violet are indicated for the gram-positive bacteria, while for the reddish or pinkish stain are the indicator for the gram-negative bacteria. So this happened because gram-positive bacteria has a thick layer of peptidoglycan. So it has the ability to retain the color, the purplish or violet color of the crystal violet that we use as a primary stain. So now I'm going to show you how it is done.
First of all, add the primary stain of the crystal violet for a minute. And then gently wash off the stain with tap water. Gently flood the smears with grams more than iodine and let it stand for 1 minute. After 1 minute, gently wash off the stain with the tap water. And then decolorizing the smears with the 95% of ethyl alcohol until the alcohol runs almost clear and leaves only a blue tinge on the smears, and then gently wash it off with the tap water. Last but not least, counter stain the smears with the saffronin for 45 seconds and then gently wash off the stains with the tap water. Blot dry the smears with the bibeless paper, and finally you can examine the bacterial smears under the oil immersion.